Hello and welcome to the 10th Longevity Snapshot, where we will take a look at a study using epigenetic reprogramming to stop intervertebral disc degeneration and reduce lower back pain. My name is Chris Linnell, and I'm the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do and how you can support us, then please check out the links below this video, where you can also find a link to the study being reviewed here. In addition, if you enjoyed this video, then please give us a like and subscribe to our channel. Lower back pain is one of the most common symptoms of aging and is a leading cause of reduced quality of life, as it can prevent people from exercising and it can limit their daily activities. Apart from injury, it is mainly caused by the degeneration of our vertebrae, in particular the intervertebral discs, which provide cushioning for our spine. There is an increase in catabolism or breakdown of tissue, as well as a buildup of senescent cells, which leads to degeneration and pain. Although pain can perhaps be managed and there are exercises one can do, there is currently no cure. However, numerous treatments are under development, and we will be looking at two of them in this and the next snapshot. In Snapshot 11, we will look at a Canadian senolytic treatment. But in this episode, it is all about the use of reprogramming. First, though, a quick recap on epigenetic reprogramming. Discovered in 2006 by Shinya Yamanaka, it allows the creation of pluripotent stem cells from any cell in our body, utilizing 4 transcription factors, better known as the Yamanaka factors. But for longevity purposes, the important part is that during the process, the cell gets rejuvenated, becoming indistinguishable from a young cell. It is therefore one of the hottest fields in longevity research, the idea that we can turn back the epigenetic clock. Recently, the process has been refined, removing one of the original Yamanaka factors, the oncogene c-MYC, which reduces the risk of tumor formation. And it is this improved version which David Sinclair used to restore vision in mice. It is also the version used in the present study, which we will now take a look at, starting with the graphical summary. The study involved rats, and they used exosomes to deliver 3 out of the 4 Yamanaka factors, OCT4, KLF4, and SOX2. They are better known as OSK, however, the authors have swapped the lettering around. So in this paper, they are known as OKS. The exosomes were taken from bone marrow stem cells, and they were transfected with cavin-2, a protein which improves the uptake of the exosomes into the cells of the animal. Once OKS had been inserted into the exosomes, they were injected into the intervertebral disc of the rats. The part of the intervertebral disc that they were injected into is called the nucleus pulposus cells, which act as shock absorbers for our spine. Taking a look at what happens inside the cells, on the left we have the aged nucleus pulposus cells, or NPCs, and a lot of them become senescent with increased DNA damage, an increase in this epigenetic marker of aging called HK20ME3. They have reduced proliferation capacity, increased catabolism compared to anabolism. So again, catabolism is the breakdown of molecules, anabolism is the building up of them. So when we increase muscle, that's a result of anabolism. And there's an increase in inflammatory markers such as TNF-A alpha and IL-1 beta, which gets released from senescent cells. Once the OKS containing exosomes gets taken up into the cells, they get rejuvenated, as shown by reduced DNA damage, a reduction in this epigenetic aging marker, which results in increased proliferation capacity, a more balanced ratio of anabolism to catabolism, and a reduction in these inflammatory molecules. Now let's take a look at the details of this study. Before moving into rats, the researchers first started with cells in culture, and in the first experiment injected aging NPC cells with this OKS plasmid, which led to a reduction in age-associated hallmarks, increased their proliferation capacity, restored their metabolic equilibrium, and reduced inflammation, thereby basically turning them young again. In more detail, looking at B, C, and D here on the left, which shows 3 markers of senescence, we can see a reduction in these markers in the treated cells. So in each of these bar graphs, the red represents young cells, blue represents aging cells, and purple represents the treated cells. And as you can see, the purple bars are similar in height to the red bars. In fact, there was no significant difference between the two. but there certainly was a significant difference between the old cells and the treated cells. E and F here show age-related stress response genes, and levels are once again much closer to that of young cells compared to the old cells. G shows a marker of double strand breaks, which is when our genomes get damaged, and the treated cells have much lower levels than the old cells. H4K20Me3 is once again an epigenetic marker of aging, and here as well the levels are lower in the treated cells. Further down here, J and L show levels of beta-gal, another marker of senescence, and once again the levels are much lower in the treated cells. In M and K, EDU is a marker of proliferation, and here levels are higher than in the old cells, much closer and in fact higher than the levels in the younger cells. N and O show levels of anabolism, and here the levels are higher than in the old cells. P and Q are levels of catabolism, and here they are lower than in the old cells, closer to that of or the same as that of the younger cells. And R and S show inflammatory molecules, and once again they are lower than the old cells and closer to that of the young cells. So this is showing that these various hallmarks of aging have been reversed in these old cells. I'll just briefly touch upon figure 2, which simply shows that the addition of this CAVIN2 protein increased the uptake of the exosomes, which we can see in G here, where the green bar is higher than the purple bar. Next, they repeated the first experiment, but this time using exosomes treated with CAVIN2 to insert the OKS into the aging NPC cells, which once again led to a reduction in the aging-associated hallmarks, an increase in proliferation capacity, a restoration of metabolic equilibrium, and a reduction in inflammation, leading to much younger NPCs. And in these bar graphs, the green bar represents these CAVIN2 exosomes. And as you can see, the green bars are the lowest for, on the left here, a senescent marker, 2 aging stress response genes, and 2 other senescent markers. In addition, at the bottom here, levels of the proliferation marker EDU were highest in the treated cells, and levels of the inflammatory marker IL-6 were lowest. I'm not sure exactly what G represents. It didn't quite explain it in the text. Now moving into animals, but starting off with some human samples in B and C with people with mild and severe disc degeneration. C simply shows that levels of P21 and P16, markers of senescence, were elevated in these patients. D shows the study design of the first treatment given to the rats. The model used to induce degeneration was by puncturing the intervertebral disc, and after 2 weeks they degenerated and became senescent. Once they had degenerated, they were then injected with the treatment or the various controls, and then 4 or 8 weeks later they were analyzed using a scanner. Now, the interesting thing about this study was that the same rat was given various treatments. So this is sort of an image of their tail, and they were either given a sham treatment, so they weren't actually punctured. They were given the OKS exosome treatment, just exosomes on their own, just OKS on its own, and sort of a saline solution. This got around the issue of variation between animals. And in the first set of results for DHI, which stands for disc height index, so how much gap there is between the actual vertebrae, either after 4 weeks or 8 weeks, and you can see that either after 4 weeks or 8 weeks, there was no real difference between the sham and the treated sections. Whereas for the 3 controls, there was a reduction in that height. And looking at the image here on the right, you can see this, the NPCs are these sort of white blobs. So that's the sham, the intervertebral discs, which weren't punctured. The OKS exosomes seem to maintain the NPCs, whereas the other 3 treated controls, this white blob sort of disappears. Here's the sort of the cross section again, white blobs here. And this was after 4 weeks, and after 8 weeks, it sort of stayed similar or the same, with the treated animals maintaining the structure. Next, in H here on the left, showing the grayscale value, which is the amount of water in the NPCs, and lower is better, showing that there's more water inside. And once again, we can see that the treated sections have no real difference, the uninjured controls, both after 4 weeks and 8 weeks, whereas the grayscale was much higher for the other 3 controls. And on the right in I here, it shows the Furman MRI score showing levels of degeneration, with 1 being lower levels and 5 being higher levels. And we can see that the treated animals, or the treated sections, show only red or blue, indicating a score of 1 or 2 after 4 and 8 weeks, whereas the 3 controls had a kaleidoscope of other colors, indicating worse degeneration. Next, we have the histological analysis of the cross-sections of the NPCs, and with the sham control on the left, the treated second to the left, and then the 3 other sort of controls on the right here. And just sort of eyeballing it, you can see the sort of white section. Again, this is the NPCs, is much similar in the treated sections compared to the uninjured controls, whereas the other 3 controls fared much worse, both after 4 weeks and 8 weeks. In B here, ACAN plays a role in the development of cartilage, and I guess the staining is much stronger in the treated animals, similar to that of the uninjured sections, compared to the other 3 controls, both after 4 weeks and 8 weeks, indicating, I guess, that there is greater potential for cartilage growth. In C, MMP13 degrades collagen. And here the levels seem to be lower than in the other controls, and I guess more similar to that of the uninjured sections. While in D here, P21 again is a senescent marker, and judging by the level of staining, the treated sections had lower levels of this senescent marker than the other 3 controls. Now comes perhaps the most important experiment, to see how this treatment can affect lower back pain. This time, however, instead of the tail being used, they punctured the lower spine of the animals to induce degeneration, and then 2 weeks later injected them either with the OSK exosomes or just a saline solution. 4 weeks later, 3 different pain tests were conducted. The first using pressure on the spine, the second using a sort of a wire on the hind paw, and the last one, the Hargreaves test, is a measure of thermal pain. And before looking at the results, here in B is a cross-section of the animal's spine, and with the sham, so unpunctured animals, you can see the NPC is quite clearly visible here. Whereas for the animals just injected with a saline solution, you can see that it is quite badly degenerated. However, for the animals given the treatment, once again, the NPCs are quite visible here, looking very similar to the uninjured controls. Now let's take a look at the results of the pain tests in C, D, and E, with red being the uninjured animals, purple being the saline controls, and blue being the treated animals. And 2 weeks after they were punctured, the injured animals showed much lower levels of pain threshold, which stayed low for the animals just given saline. However, there was a significant improvement in the animals given treatment, as you can see by the rising blue line. For the other 2 measures of pain, however, there was no significant difference between any of the 3 arms. Now, the researchers think that this might be due to the fact that the puncture did not rupture the outside of the disc to cause herniation, and did not stimulate the nerve root to cause pain. Now, in intervertebral disc degeneration, the nerve endings expand because of an increase in nerve growth factor, which leads to increased pain sensitivity. And this hypothesis was partially corroborated by the fact that nerve growth factor did not increase in the treated animals, as we can see here in F. In these images, the red dots represent the nerve growth factor, and the saline treated animals had a lot more of these red dots compared to the treated animals or the controls, which we can also see in the bar graph on the right-hand side, where again, this is showing nerve growth factor, and the treated animals were much closer to the uninjured control animals compared to the animals just given saline, with this large blue bar here. Lastly, the researchers also looked at the transcriptomics, and without going into the details, they found that 215 genes were upregulated and 142 genes were downregulated, and those mostly sort of involved an increase in anabolic genes and a decrease in catabolic genes, indicating that there was more growth than degeneration following treatment. And that marks the end of the study, ending with the graphical summary once again. So although it wasn't a complete slam dunk in that only 1 out of the 3 pain tests saw an improvement, however, that's probably more down to the study design. What is clear, however, is that the old and degenerated cells were rejuvenated to a certain degree, and that visually the treated MPCs had the same appearance as the uninjured ones. It's also great to see another example of cellular reprogramming in live animals, as there have been relatively few published studies to date. It's also interesting that they used exosomes for the delivery of the Yamanaka factors as opposed to viral vectors, which might provide some additional benefits. And although they were injected directly into the disc, that delivery method could be used systematically to deliver the payload throughout the body. All in all, epigenetic reprogramming shows great promise, and this is yet another example of that. Let me know what you think about this study in the comments below. And with that, we've reached the end of another longevity snapshot. If you've enjoyed this video, please give it a like, share it with your friends, and subscribe to our channel. In the next episode, we will take a look at another way to reduce lower back pain, this time using senolytics. So until then, goodbye.